So hello everyone. I hope everyone's doing well. So I did my presentation on the paper Global and Local fMRI Signals Derived by Neurons Defined Optogenetically by Type and Wiring. This paper was by Lee and colleagues, and I'm going to discuss it further now. So a little background before I delve into the paper itself. Prior research has not categorically observed the activity of a set of neurons can cause bold signals. Bold signals, as I will talk more about in another slide, is short for blood oxygenation level dependent. And the theory behind it is that when you're using a particular region of your brain, let's say you're observing an image, then more activity would be seen in your occipital lobe, and therefore more blood and oxygen will be needed for that region, and that can be picked up on an fMRI image. Overall, prior studies have not been able to demonstrate this when local excitatory neurons are activated. In addition, when MRI-compatible electrodes are used for stimulation, there's random activation of neurons, be it local excitatory, inhibitory, modulatory, as well as anti-dramatically driven non-local cells that happen to have axons within the stimulated region. So you're not observing the activity of the certain neurons that you're specifically examining. To fix this, the researchers decided to combine optogenetic stimulation, which I will cover further in another slide, with fMRI readouts, so you can be able to stimulate a specific set of cells and observe if this causes a signal in the fMRI image. So the overall goal of the paper was to examine if activation of local excitatory neurons using optogenetics can induce bold signals in a particular region. There were two main techniques that were integrated to conduct the study. The first technique was bold fMRI, which is short for blood oxygenation level dependent functional magnetic resonance imaging. And this technique is used non-invasively to note brain activity. As I talked about previously, these bold signals assist in being able to delineate which parts of the brain are active during a certain activity. And they rely on three aspects, which are cerebral blood flow, cerebral blood volume, and cerebral metabolic rate of oxygen consumption. The other method that was also used was optogenetics. This technique allowed the experimenters to stimulate a specific set of neurons by using light to control the neurons that have been genetically modified to express light-sensitive ion channels. So by combining these two techniques, the researchers then conducted the experiment. The animal model they chose were adult rats, and these animals had a viral vector inserted into their primary cortex, the M1 region, to drive CAMK2 alpha expressing neurons. Also, I would like to add where the virus was injected to drive the expression of channel rhodopsin 2 was also the optical stimulation site. They used 473 nanometer light pulses at 20 hertz with 15 millisecond pulse width to stimulate the targeted cells. So before I go on and talk about the results, I want to note that the experimenters tested if it was feasible to obtain fMRI signals from the animals and made sure that when they used optogenetics, they were stimulating the neurons that they specified. Once they observed everything was up to standard and the study could be done, that's when they went on with the experimentation they had intended. The first aim Lee and associates had was to note if optogenetically stimulated neurons could showcase a local positive bold signal. And the results showcase that optically evoked bold signals were seen in the experimental animals, but not the control, which were given a saline solution instead of the opsin viral vector. These results show that using optogenetics, a specific set of neurons can be triggered and their activation can be seen on an fMRI image. The figure I have here is from the paper, and on the left you can see that there were no bold fMRI signals for the control animals, but for the animals that had the vector inserted, they clearly showed a significant signaling. Since the investigators were able to exhibit that optogenetically stimulated neurons show bold fMRI signals, they next wanted to expand this knowledge and look at microcircuit properties using this technique. So they inserted a viral vector in the thalamus and looked at the expression of cortical thalamic axonal projection fibers. Here, they observed strong thalamic bold signals in response to the stimulation of the primary cortex. primary motor cortex, but the signals they observed were unusual and different from what they saw. And when they looked at the cortical neurons, for example, they saw a delay in spike rate increases in the thalamic neurons. So if the unusual kinetics they observed in the thalamic bolt signal, Lee and colleagues wanted to note if the delayed thalamic bolt response is similar with local thalamic electrical activity. And in doing so, they assessed the thalamic response using simultaneous optrode stimulating and recording in motor cortex and electrode recording in thalamus. So they didn't use optogenetics here to stimulate the cells and instead directly triggered the neurons and wanted to look at the response. They observed a similar pattern in direct recording of the thalamus as when bolt signals were observed. So no discrepancy was seen between thalamic bolt response and local thalamic electrical activity. So their work could be continued. The final aim they had was to examine the connectivity arising from initial drive of anterior posterior thalamic nucleus projections using OFMRI. The prior connectivity they looked at was based on the connectivity of genetically similar cortical and thalamic neurons. Now they wanted to observe the connectivity of neurons by topology. As previously mentioned, electrode-based stimulation is not very specific, so electrode stimulation can drive both antidromic as well as orthodromic projections, so you can't really distinguish between cortical thalamic versus thalamocortical projections. However, the new method used by researchers was successful at representing the projections from posterior and anterior thalamic nucleus to respective destinations of the cortex. For example, stimulation of posterior thalamic nuclei resulted in bolt response at the ipsilateral thalamus and somatosensory cortex, while stimulation of the anterior thalamus produced signals in the ipsilateral thalamus and bilateral cortex, which is consistent with known connections in neuroanatomy. One thing that I found notable, however, was that the researchers wanted to note the projections from the anterior and posterior thalamus to the motor cortex, as they stated that thalamic projections to the motor cortex would be more likely than those to the sensory cortex to involve both ipsilateral and contralateral pathways, since motor control and planning must involve bilateral coordination. But they looked at projections from the thalamus to the somatosensory cortex. Now, I'm not sure if they initially looked at projections from the thalamus to the motor cortex and did not get the favorable results or what happened, but they didn't really expand upon this and why they had the initial goal of looking at pathways from the anterior and posterior thalamus to the cortex, but they stated results to the sensory cortex. I thought it was strange, and as a reader, I would have liked for them to delve a bit more into this, but I guess we just gotta take their word. So this figure demonstrates the prior results I was talking about. The top is the image of the signaling observed in the somatosensory cortex when the anterior thalamus was stimulated, and you can see that signaling is very strong ipsilaterally. On the bottom are bolt signals from when the posterior thalamus was stimulated, and you can see both ipsilateral and contralateral signaling. So the overall data gathered showed that OFMI displays clear bolt signals when a specific set of neurons is stimulated. Also, the technique allowed for the mapping of the causal connectivity of genetically defined neurons in specific brain regions, and it allowed for the causal connectivity mapping of cells defined by circuit topology. We just got an email from her saying that we're continuing without her. So my own thoughts of this work are that it was really intriguing, mostly because with this technique, you can possibly map intact neural circuitry. However, I think this research is more beneficial for basic research and clinical studies. And a lot of you guys' questions were asking about clinical basis of this research, which I'll get to when I get to the question section. But I do think it's mostly for basic research. But with advances in optogenetics, I think maybe it may have benefits in medicine as well to study neural circuitry disorders in the future. I believe future studies can use the technique to look at more complex brains, such as non-human primates, and can maybe analyze more complicated connectivity. So the paper was from 2010, so I did some research into current work using the same methodology. I found a paper from 2018 which used OFMRI to examine the downstream forebrain regions modulated by activation of a region of the cerebellum that induces stereotypical ipsilateral forelimb movements. This research was able to show the connectivity map of discrete regions of the cerebellar cortex, functionally projecting to the forebrain using OFMRI. Another study from 2016 also used this technique to investigate whether multivariate dynamical system models can accurately estimate causal functional interactions between brain regions. Overall, the studies using the technique that I found are mainly using it to be able to map out neural circuitry of different regions. With that being said, I'm going to move on to your questions. So the first question is, do you think something like this can be done in humans? Do you think there will be any ethical issues? Give me 1 second. So I think OFMI is currently more useful for animal models than humans. I do believe in the future optogenetics can be done in human subjects as well. However, the technique can potentially be used in human trials, but there needs to be more trials and advances before doing so. Now, the ethics of it can be a little tricky to deliberate on, but if it seems safe and a subject volunteers, then I do not see any ethical flaws in performing it. Second question is, since this paper has been published, has this approach been used to map the global effects of controlling a local cell population in mammalian brains? Since the publication of the paper, various studies have used the same methodology in more complex species. For example, doing some research, I came across a study that used optogenetic fMRI within the living and intact mammalian brain to reveal bolt signals in downstream targets distant from the stimulus. Other studies have used the technique in mammalian brains as well. The third question is, in this paper, it concludes that BOLT can be used to search for global circuit disease endophenotypes. Have there been any findings since this article was published in 2010? So there have been numerous instances in which BOLT signals have been used to map out global circuit disease. Neurodegenerative diseases are one example, as the synaptic activity does not correlate with the cerebral blood flow due to synapses being disturbed. Also, behavior and brain disorders can be observed using BOLT as well, since they involve neural circuits, and you can use BOLT to map out functional connectivity of disturbed circuits and assess how this connectivity evolves over time. Since the publication by Lee, there have been many studies that use OFMI and apply it to observe different circuits and any abnormalities in them. So the paper has really been pioneering in the scientific community and allowing others to use the technique for their own goal. So next question is, what do you think any future uses of this technique will be, or where can this technique bring us? So I believe that future uses will encompass looking at different neural circuits and connections in basic research. But as I stated before, maybe if advancements in optogenetics, it will become a tool to be used in clinical studies and possibly looking at different global circuit diseases. Next question is, do you believe this technique could have more clinical uses, or does unknown signal triggers leave this to be strictly research oriented? The answer for this question is similar to the one before. Clinical uses can only come after ensuring that optogenetics is safe to be conducted on humans. What challenges do you think they would face if this study was performed on humans? The biggest challenge would be the optogenetics part of the research. fMRI is commonly used in clinical settings, so I don't see that being a part of the obstacle. And the last question is, how do you think fMRI is useful now? Flash forward 10 years from this article. So fMRI is useful because it allows for estimation of the regions of the brain that respond to sensory stimuli or to tasks, and is also useful for detecting regions of the brain which show functional connectivity. So with that being said, that's the end of my presentation. Thank you for your time. I was going to ask, does anyone have any questions for me? But I don't think anyone does. So yeah, I guess that's it.